I'm going to start by acknowledging my collaborator who pulled me and Susan into the autism field about some 15 years ago. Of course, the funding agency and also in particular some of our trainees, among a long list of very successful past and present trainees, in particularly Ting Wang, Ashley Scott Van Zeelen, and Jeff Rudy, whose work I'm going to be telling you about. And of course, all of the children and their families, because without them, we wouldn't be here telling you about our findings. I have a lot to cover, so I'm going to just jump in and give you a 30-second spiel on fMRI. Of course, by now, most of you know, this technique lends itself to be used with children beautifully because it's completely non-invasive. It allows for repeated testing, repeated scanning, so you can track development as it occurred over time. It is very safe for young children and indeed for babies, as you'll see later on. This is one of our subjects. By the time we are ready to make them go inside the scanner, they are not even aware that they're going into that narrow tube because they have a nice setup, some miniature goggles. They display the videos of their choice at the beginning, and then whatever it is that we want them to see. They have nice headphones and a mic so that we can communicate. So they're pretty comfortable in there. And most of what I'll be telling you today is about brain function and also functional connectivity, which is in contrast to what traditional standard structural MRI tell us, which is, looking at how the brain looks like, not about what it does or how it may be wired. Okay, so this would be the topic. There are a lot of studies looking at structural brain imaging autism, but it would not be possible to cover them all in one talk. So a mini tutorial on the social brain, or at least on a few of the regions that I'll be telling you about in a mid-sagittal section. So imagine cutting through the nose. Here you see some of the usual suspects in the reward circuitry that include the nucleus accumbens, the ventral striatum, and also the orbitofrontal cortex and the amygdala. And so the other set of structure that I would like to highlight for you are these regions which have been, or are part of a network that it's important for mentalizing. So thinking about others, but also about self-other distinctions, information about yourself, and basically social cognition and interpersonal understanding in general. Of course, there are several other social brain regions that have evolved more recently, and I'm just kidding here. I should add a few, actually. I have 16 years old almost, and she knows now about, she's using Instagram and other things like that. But more seriously, there are several other regions now. We're looking at the surface, basically on the outside of the brain, and there are several other regions that have been implicated in social cognition, the temporal poles here, as well as the superior temporal sulcus, and also the temporal parietal junction, that are all important in processing socially relevant information. In addition to that, over here you have your language areas. Of course, autism shows deficit in language, so those are also areas that have been popped up and been examined in a huge now literature on autism over the past 15 years. That have been hundreds of studies that have tried to characterize the functioning of these many regions that I have briefly described to you. And if I have to summarize it in one sentence or two, I would say that most often than not, the studies have reported hypoactivation, so less activity in many of these structures, in mentalizing regions, in language-relevant cortices, as well as in other regions that process socially relevant information, such as the famous fusiform face area, which is an area that, as you may guess, is very it's very important for processing information from faces. However, after a few years, we also started noticing that there were several studies that actually reported normative activity, so similar level of activity in the same regions in individuals with autism, if you tweak with either the type of stimulus. For instance, if you were to present individuals with autism with pictures of their mothers or some people they were familiar with as opposed to complete strangers. Or if you did something very silly like putting a fixation cross at the level of the eye region, so to direct the attention to this very important portion of the face. So with one of my first graduate students, we decided to test more formally the notion that some of the differences that we have been seeing in the first wave of studies, that is the reduced activity or responsivity in the system, that it may actually have simply reflected of the fact that the individuals with autism don't necessarily pay attention to the same thing or to the stimuli that we presented them. And so she came out with a very clever paradigms. Indulge me, if you will. It took forever to draw these nice cartoons and to record the stimuli. So our kids in the scanner would be presented with a scenario like that, and they would hear something like this.
Brian and Dina are blowing up balloons.
And then they would see another one of these pictures, so they could be like this.
Brian keeps blowing his till it's huge. Dina says, nice going.
Or,
Brian keeps blowing his till it pops. Dina says, nice going.
and then they would all be asked.
Did Dina mean what she said?
Okay, and so what we did, we had several of these vignettes, and any single child would either see one scenario or the other, never both of them. And so in the first block, when they were being scanned, we just presented a bunch of these scenarios and asked them just to answer basically the question, whether the speaker was sincere or not. And in the second two blocks, we either told them to pay attention to the kid's face or to pay attention to the kid's tone of voice. Okay, and so what did we find? We actually did find evidence that attention does indeed modulate brain activity. So this is when we did not give them any specific instructions. As you can see here, the typically developing, abbreviated with TD throughout the talk, shows significant activity in one of those regions that I've highlighted earlier, one of those theory of mind regions, okay? And you don't see any similar activity in the children with autism. But when you're looking at the conditions where we did warn them, pay attention to the kid's face or to the kid's tone of voice, now both groups show significant activity. And for those of you who are statistically inclined, let me tell you, it wasn't just that it was just barely above our threshold here. There was actually a significant interaction in this region, basically showing that there was a significant difference here and that that was not longer the case there. And I want to point out something important here, and that is that this differences in activity did not have anything to do with behavioral performance, which was very high in both in terms of accuracy as well as in terms of how long it took the subject. There were no group differences whatsoever. And if you're worried about like, wait a second, so how could they do the task? I mean, so, well, if you think about it in this particular scenarios, they had a lot of information available. They knew that something bad or something good had happened, so they could have just relied on some sort of strategies where if I know that a balloon is popping to somebody's face and the person says, way to go, that is, no, the person didn't mean it because you have a positive statement with a negative outcome. But in everyday life, many times you don't have the luxury of having this redundancy in information. You meet with a friend for lunch and say like, oh, how are you? And they tell you, oh, I had such a great day. You know, you don't know, you don't need to know anything. You would have that information available to you. But the point that I want to, the take-home message I want to make about this slide is the notion that we should really be careful about making inferences about the integrity of any brain system just based on behavioral performance. Sometimes it's been done in the literature. You say, okay, well, you know, behaviorally there is no differences between my children with autism and the normal, so since we know that this behavior is typically subserved by this brain network or structure, then there must be nothing wrong with that brain network or structure. But that need not to be the case, because there can be compensatory mechanism at the brain level that allow you to perform a task relying on alternative circuitry. Okay, so why this lack of an implicit or automatic attention for the human face and the human voice that we have seen, and not just in the imaging studies, there is actually pretty extensive behavioral literature that shows that. And so here's where the social motivation hypothesis comes into play. And according to this hypothesis, individuals with autism, indeed young babies that then go on to develop autism, fail to show a preference for the human face or for the human voice because they're not intrinsically rewarding to them. Okay, well, this is nice, but you cannot really test this hypothesis behaviorally because you get caught in a loop. Well, they don't pay attention to faces as much. They don't prefer the human voice. Why is that? Because it's not rewarding. How do I know that it's not rewarding? Because they're not looking at faces, because they're not preferring the voice. You see that it doesn't work well. But thankfully, we can study the reward circuitry in the brain in individuals with autism. And that's a circuit that has been very well characterized. And so with another student, Ashley Scott Venzila, we set out to do just that. So we examined the neural correlates of reward processing using a probabilistic task and using both social and monetary reinforcers. So this is kind of like a busy graph, bear with me. So they're just seeing this kind of pretty fractal images and one at a time, and they're told that they have to guess whether this is a 1 or a 2. And they're told that it's kind of hard and the computer messes up, so that they basically have to guess. But in reality, there is implicit learning that it's happening because we're giving them feedback. And the feedback that they're getting, it's either in the form of a social reinforcer with a very pretty girl smiling saying that's right, or a pouting face, that's wrong. You see them here. We also have some control condition with a neutral face, or they knew that they were getting money when they got it right, or no money. And what did the data show? Well, as you can see here, you see a very strong activity in the typically developing subject when for both type of reinforcer. And you don't see significant activity in the same ventral striatal region that I mentioned to you as being at the core of the reward circuitry in kids with autism. And when we did the direct comparison, because statistically you have to do that or else you can't say much, or you cannot publish your findings, you do actually see that there was a significant difference, again, in the ventral striatum and also in the anterior cingulate, meaning that the typically developing children show stronger responses in the reward circuitry for the social rewards, okay? Not so much. So the difference for the monetary didn't reach significance. Okay, and just to prove maybe something obvious to you, I just want to show you a study that came out right after that that showed that live face-to-face interactions during fMRI actually leads to increased activity in this reward circuitry. Now, you may say you don't need an fMRI study to tell you the social interaction is rewarding, but actually the study made a profound point because we're not talking about being in a bar, talking with your friends and having a good time. These people were in a scanner and they were just interacting with an experimenter over a computer interface. So it wasn't really like, whoa, I'm having a party. And nevertheless, the reward circuitry lit up to a significant greater extent when this interaction was live as opposed to when it was a recorder and they knew that the responses they were getting were just generated by a computer. Okay, so please feel free to stop me if I am mumbling, going too fast. I mean, I'm just trying to catch up and so that we can get to the wrap up and in time. Okay, so now the human face and voice and social interaction may not be intrinsically rewarding to individuals with autism. The question then is, why would that be so? And the answer is not a definitive one, but I'd like to propose to you that it may have something to do with mirror neurons. Now, I don't know how many of you may or may not have heard about mirror neurons. They became kind of hot in the press for a while, and also they've been subject to some controversies, but let me tell you just very briefly, they were first discovered in the monkey because they were discovered during single-cell recording, so you need to have an electrode implanted in the cortex of an animal to do that. And the property that this neurons display is one where they fire, as you see here, both when the monkey is performing an action as well as when the monkey is observing somebody else doing the same actions, okay? And so, I mean, to make a very long story short, the system is thought to provide a neural mechanism by which we can understand the action and intentions of others in a very automatic, implicit way. So right now, if you see me reach for the bottle of water, I mean, a no-brainer, you know what I want to do, right? I want to drink. And why? Because I'm thirsty, or you may think because I want to think about what I'm going to tell you next. But, you know, it gives you kind of like an automatic reading. And when I look at your faces, if I see you going like that, I'm going to know right away you didn't catch what I said. If I see you smiling, I'm thinking, okay, they're with me. Okay, so we are constantly fed this information during our interactions. And the notion here is that the system then, if it's disrupted, can really cause a lot of the problems that we see in autism. You know, would make really social interaction very hard if you had to decode a facial expression and learn that, you know, when people frown, it means that they are angry and so on and so forth. Okay, so there's also been recent evidence that mirror neurons do exist in humans, and not very surprisingly. But what I want to make, the point that I want to make here in showing you this is that this single cell recording in humans cannot be done at whims. You know, you cannot just go and record in the same area of the brain where we found them in monkeys. You have to do it based on the need to do it, this intraoperative recording. So these were pre-surgical, these were surgical patients we're talking about. So the interesting fact is that they found that there were neurons that display this mirroring properties in regions other than the motor regions that they were found in monkey, which is consistent with this growing evidence. This mirroring is not just a peculiar property of the motor system, so it doesn't work just when you see somebody performing an action, but that this vicarious activity can actually help us understand also other people's emotion as well as their sensation. And I mean, I don't have it here, but if you've seen a movie where there's one of my colleagues always used this clip of a James Bond movie where there is a black tarantula crawling up on James Bond while he's asleep. And when you are in the movie theater and you watch it, you're kind of like, it's almost like you can feel it, you know? Okay, that would be an example of what I'm trying to convey here. But now back to why I see that there could, why I'm trying to make a connection between mirror neurons and social attention, is that we know from some of the early abilities that both newborn babies as well as monkeys shows from the very early few actually days of life, is that some of this mirroring seems to be happening at birth, at least for some of the very basic rudimentary motor actions, if you will, that babies have learned to perform, basically with the orofacial musculature. And so the notion here is that perhaps the ability to understand other actions by being able to map, you know, a mouth open, a tongue protrusion onto your own motor repertoire may actually give rise to this preferential attention to other human beings. Okay, this is a system that since our first study, no, actually it wasn't the first study, but since we did a study that received, I guess, a lot of attention, has been characterized in over 24 studies, and I'm going to tell you about the studies that we did where we looked at mirroring of others' emotions. So the kids in the study were being presented with a variety of different emotions, and they were either told to just look at the expressions on a person's face or to imitate that expression. Because what we know is that the system is important for imitation in addition to social cognition and the other things that I mentioned earlier. So what did we find? We found that the typically developing kids, as well as the kids with autism, perform the task. How do we know? Because this is motor cortex activity, so they were imitating. But only the typically developing children had a lot of activity in this mirroring region, the frontal component of the mirror system. And when we did the comparison, again, between the two groups, that's exactly where you saw the significant differences. Now, I want you to know that, I mean, the kids with autism were doing the task quite well, and this is one of our subjects. And I think that even before I clicked to the next animation here, that you could tell that this kid was here imitating a person making an angry face, a happy and sad, and so on. So they were doing it quite well, and indeed we test this empirically. We had blind observer code the imitation that both typically developing kids and kids with autism did, and they could not tell them apart, even if our observers were people that were quite familiar with and working with children with autism. Okay, so how did they do it? Well, going back to the point that I mentioned earlier, there can be additional neural strategies that you bring on board, and indeed, this is where the kids with autism show greater activity than their typically developing counterparts. And these are two regions in the visual association cortex and in the parietal cortex that are important for visual motor attention. So the notion is that the kids could actually do that imitation task quite well by paying extra attention to what they were seeing and translating that into a motor plan that would produce the same facial expression. So they had to do it this way because they didn't have that implicit mechanism going for them. And we recently replicated this study in a much larger sample. Here, just showing you the direct comparison straight on. I'm sorry, it's a little bit dark. But you can see again that the typically developing children had significant greater activity in this frontal component of the mirror neuron system. And this is looking at a sample of 40 children, which in comparison to social studies may not look very large. But in terms of imaging, it is very large. Typically, you have imaging studies in the teens or in the mid-20s and so on. But what was important for us to look at this further, because in our prior studies, children had to have a clinical diagnosis that was confirmed by both the ADI and the ADOS, okay? I don't know, does anybody here not know what the ADI, the Autism Diagnostic Interview, and the Autism Diagnostic Observation Schedules are? I think not, but that's what we're talking about. And so in this larger sample, we actually still have a clinical diagnosis confirmed by ADI, but then some children came as having full autism on the ADOS, some children only were on the spectrum on the ADOS, and some children did not come out at all as being on the spectrum. And look at what happened. When we are looking at the kids that had a full autism diagnosis on the ADOS in addition to ADI, you see that the differences are even more striking than what you see when comparing across the whole sample. And indeed, again, for the statistically inclined, the differences were stronger. They survived a higher statistical threshold. Now look what happened if I am now comparing the typically developing kids to the subset of kids that were more mildly impaired. They only were on the spectrum, or they did not come out on the spectrum at all. Now the differences have almost disappeared, and they would not survive the most stringent threshold we typically apply to the data. So we need to be very careful about the heterogeneity in the samples of research studies, because I think unfortunately, a lot of the discrepancies in the literature may actually be simply arising by slight sample characteristics, difference in sample characteristics. We pushed this one bit further, and we actually used we ran a regression analysis with the ADOS social subscales, because we've also used it in the prior studies. And basically what we did, we look at the score on the ADOS social subscales, and we say, where is activity modulated or vary as a function of how impaired a child is? And lo and behold, look at the very strong relationship that you see in this area. This is across the whole sample. And then I've also plotted it here just to make it easier to follow, hopefully. So you know that obviously lower score means that you're less impaired. And in red are the kids that were only coming out on the ADI. In orange, the kids that were on the spectrum on the ADOS. And then the most impaired kids in yellow, you can see where the kids that had autism on the ADOS. So it's a very nice, strong linear relationship there. Okay, so the next question, say that we buy into the notion that this mirroring mechanism is not functioning as well in individuals with autism. Why would it be? And I'm borrowing from the media this notion that maybe, you know, if they're not broken, maybe they're fogged up a bit. And the answer may come from studies that have shown that connectivity in individuals with autism, that the brains of individuals with autism may not be wired quite as typically neurotypical controls. So in recent years, there have been large bodies of studies that have used both functional MRI as well as other technique like diffusion tensor imaging to look at connectivity in the brain. So this would be like how we're looking at functional connectivity, and this is a DTI image. And they've shown typically reduced cortical functional connectivity and also reduced white matter integrity. This would be kind of like structural connectivity that we're talking about here. And indeed, it's also been postulated that there are some studies that also found stronger connectivity, but typically between subcortical and cortical structures. But overall, the hypothesis has been put forward, I think Dan Geshrin alluded about earlier, that autism may be viewed as a developmental disconnection syndrome, and that early in development, there may be an overabundance of local connection. They may actually interfere with the establishment of long-term connection that are typically happening in the normal brain here. Okay, so we have done some work in this area, and in particular, since we were interested in that mirroring regions in the pars superciliaris, we focus on this region, and what we call it a seed. Why? Because that's what you do when you're looking at functional connectivity. You start with a region, you extract the time series. So you would have, you know, like you're basically during the course of the scan, you see how the signal goes up and down in that region, and then you're saying where else in the brain, what other brain regions show an activity pattern that is correlated with the activity in this region. The notion is that two regions that are working together to perform, say, a task would have a pattern of activity that tends to oscillate or, you know, change together. Of course, you can also see the opposite. So you can find regions that show the opposite pattern. So just like as when activity goes up for this region, there may be other regions where activity typically goes down. And so that's what you see, oops, sorry. That's what you see here. So in the yellow-orange color, you see the regions that were positively correlated, so the regions that show patterns of activity that was in sync with the activity in this region. And in blue, in the cold colors, you actually see region that there was the activity was inversely correlated. And overall, your eyes may be struck by the similarities that we see in between the two groups, but if you pay attention, there may also be some differences that may pop up to your eyes, and to make it a little bit easier, I have highlighted them here. So this is where the two groups differ significantly, and basically this is a region where the typically developing controls show stronger connectivity. So this region here was more strongly in sync with this region in the typically developing control. And that's important because this is actually a region that also is implicated in this mirror neuron system. There were also regions where the children with autism show stronger local connectivity. Again, this is a frontal region, right? So this posterior region here would be increased long-range connectivity. You're talking about a frontal brain region talking to a parietal area, whereas here you're staying within the same frontal region. So consistent with the model that I mentioned earlier, the typically developing kids seems to have stronger connectivity with long-range regions, the kids with autism, stronger connectivity within, and now I may confuse you all together, but there's also stronger negative connectivity without the frontal region in the typically developing. The take-home message is just corroborating evidence that this mirroring region is not as strongly connected with parietal regions that are also involved in the similar in the same system, and also corroborating evidence for the notion that there are significant alterations in the connectivity in the brain of kids with autism. Now, more recently, we have been looking at connectivity that it's independent of a particular task. What I've shown you so far was actually connectivity between regions that we were comparing activity when the children were looking at those emotional phases. What has been done recently is to look at what has been termed resting state fMRI. So you put your subject in the scanner and you tell them to basically do nothing. So you tell them to rest. Sometimes you show them a fixation cross and you tell them to look at that. Sometimes you tell them to have their eyes closed. And you're just relying on synchronized low-frequency spontaneous fluctuation in neuronal activity, which are occurring across different brain regions. Okay, now what you see here are several different intrinsic connectivity networks that have been identified. So basically, in a nutshell, the notion here is that regions that tend to subserve similar functions tend to activate or oscillate in activity together. Okay, so you have some of the basic sensory system, like the auditory areas, the visual areas, or the somatosensory areas, but you also have more higher order networks such as the control system and the dorsal attention, and then the fault mode network I'll tell you in a minute. But the point here is that you can see beautiful activity in auditory cortices, but they were not processing any auditory information. This is just a way to basically detect how the brain has worked together in the past and has organized itself in meaningful kind of unit, if you will. So this intrinsic connectivity network are found whether a person is asleep or even when they're under anesthesia. So this is beautiful because you can now apply this strategy to study the brain of even lower functioning in children that may not have been able to perform any task while they were being scanned, or also in babies. And as I said, this intrinsic connectivity network, basically we take them to reflect the long history of co-activation between brain regions subserving different functions. So in a way, they are a way in which we can tap about how what the brain has learned, how the brain has functioned in the past. So imagine like a trail, you know, through in a forest. I mean, when something has worked, now you see a connection between different parts, different places where people wanted to go. Okay, so what do we know about how this connectivity networks develop? Well, there is a lot of work that is beginning to be done, and I cannot review it all, but I just want to give you 2 main points. One, when you're comparing children to adults, you see an increasing integration within networks. So forget about all of the names and the labels on this one. Just, I hope you can glance that there are many more connections in the adults that you see between the nodes of this network in children. And the second point is that there is actually also increasing segregation between different networks. So you see here that in the adult, now these two networks are completely segregated. There is no point in common, whereas in the children, you still see some nodes that kind of link the two together. So now I'm really going to try not to annoy you with busy slides. Please do not try and read all of this tiny little thing in red. You can even disregard these labels because I will not have the time to talk about test positive, test negative network, but I hope that you can see one important point, and that is that when comparing the typically developing children to the children with autism, that you see that these two networks are more highly segregated. There's only one point in common here between these two, where there are several points in common. So the real take-home message here is that when comparing typically developing children to children with autism, we see something that mirrors the difference that we see when we're comparing neurotypical adult to neurotypical children. And so one of the important questions that the field has to address is to what extent the differences that we have been seeing when comparing individuals with autism, children with autism, to typically developing counterparts, what of those differences may really reflect either a causal mechanism or a delayed pattern of connectivity versus just a delayed pattern of connectivity. The brain is plastic, experience, shape, brain activity and connectivity, and so to a certain extent it almost is, it's intuitive, right, that the brain of a child that has not nearly spent as much time interacting with others, talking to others, it would be less connected, perhaps looking more like a younger brain in comparison to a typical developing child. Does it make sense? Okay. All right, so, well then, you always have to move to the next level, and so now the question is, what may cause this altered connectivity? And I would just say, like, well, let's blame the genes. That's kind of like an easy, straightforward answer, because we, of course, know that autisms are very highly heritable, and then clearly stress that. And while we have begun to identify a lot of this autism risk genes, we still know relatively little about about the mechanism by which genes impact cellular pathways, microcircuits, all the way up to functional and structural brain networks and ultimately to behaviors. But we know there are studies that have shown that pattern of functional and structural connectivity are actually heritable. And so we started to look at a few candidate autism risk genes to see how they may impact functional and instructional brain networks. So one of these genes is the catnap. I like to call it catnap. We actually compared risk carriers versus non-risk carriers, and they differ in terms of brain activity in this medial prefrontal region. This is a weird inflated brain, so maybe you don't recognize it, but it's always that usual suspect. And then we took this region, as we treated as our seed region, we extracted the time series, and we basically looked for wherever else in the brain there may be activity that co-varies with this one. And we found that the non-risk group had significantly stronger connectivity between this frontal region and this posterior region. So increased long-term connectivity in the non-risk and increased local connectivity between this frontal region and other frontal regions in the group that had the risk allele. The point that I want to highlight here is that this was the pattern that was observed in both typically developing children as well as in children with autism. So we can't quite say that this is what causes autism, right? It's actually one of those risk factors. It's a vulnerability, but we still unfortunately cannot, pinpoint this altered connectivity as being the cause of the disorder. We have now, Rudy has been done an excellent series of studies looking at another important autism risk gene that has been ranked #2 in a recent SFARI ranking, I guess, of gene associated with autism. And this MET is a key regulator of signaling pathways for neuronal migration, axon formation, and synaptogenesis. There is a lot of information, a lot of studies that have linked this risk gene to autism. So for instance, in postmortem studies, a reduced MET protein has been found in the temporal cortex of individuals with autism. And the common C allele, which is been found to be associated with the autism risk as compared to the G allele and across a number of studies. And importantly, this risk allele has also been associated with increased social and communicative deficits in individuals with ASD. So in one, I only will tell you about one of his studies, and that's the one where we focus on the default mode network connected and in relation to MET. And so this network I mentioned earlier is one of those higher cognitive function network that it's involved in social cognition and theory of mind processing. And it's one where there's been perhaps the largest number of studies so far finding altered connectivity in individuals with autism. That's why we decided to focus on this one. We used resting state of MRI. Our subjects were told just to rest and not do nothing. We had a fairly large sample size here, and our group were very well matched in terms of a number of variables, including head motion, which is important when you're doing scanning. We chose the posterior cingulate, which is considered the hub of this network, as our seed region. And again, we then look at everywhere else in the brain for a region that may be connected. And as you can see here, typically the anterior, you see in the medial prefrontal cortex, I mean, this is another very important hub, as well as some parietal regions. So what do we see? This is the non-risk group, and here you see the risk group. This is sum across diagnosis, okay? Again, the pattern looks similar, but I hope you can see that there seems to be less connectivity, or the strength of the connectivity seems to be reduced between this parietal seed here and the frontal hub. When we do the direct comparison between the group, thankfully, we do find that there were significant differences in these regions. So then we zoomed in this region to characterize a little bit more carefully the pattern according to both risk group as well as diagnostic status. Okay, so this is the region, and what I'm showing you here, we extracted from this region parameters that told us that index the strength of the connection between this region and that posterior seed. And here's what we see when we are looking at the intermediate group that only had one of those risk alleles. As you can see, I mean, there was a significant difference. So the risk group had I guess, least well connected, and the group that the non-risk was obviously, as we would have expected, the most strongly connected, and the one in the middle was really like an intermediate place. When we're just now summing across the risk status and just look at typically developing kids and kids with autism, we see that there is a significant difference, such that the typically developing kids had stronger connectivity than the kids with autism. So then we unpacked it one more level, and now we're seeing the three risk groups, subgroups, split by the two diagnostic groups. And here's what it's interesting. It's very interesting because, okay, both the typically developing kids and the kids with autism show a significant difference between when they had both risk allele and when they didn't, but the intermediate group acted quite differently between the two groups, meaning that in the typically developing group, it was I'm sorry, I should have not animated that. There was no significant difference between having one or two, whereas in the kids with autism, just having one risk allele, lowered the connectivity significantly. I mean, I know this is a lot to take in, but basically the notion here is that if you have autism, just one risk allele, it's going to impact your connectivity to a higher degree than it does if you are instead typically developing. So obviously the big question there is like, what other factors are important there in determining the fact that you're taking a bigger hit if you already have autism. Okay, the last autism risk gene that we have been focusing in collaboration with Liana Hernandez is the oxytocin receptor gene. And again, in terms of background, I'm going to go very fast because I want to get to the data. We know the oxytocin is strongly associated with affiliative behavior. It's densely expressed in the nucleus accumbens, which I hope you remember is at the core of the reward circuitry. There are a lot of several oxytocin polymorphism that have been associated with autism, and one in particular has also been linked to both increased risk for autism, as well as with reduced neural activity in this Salience network. It's another one of those intrinsic network, one that I've not talked to you about so far, but as the name kind of implies, it's a network that it's deemed to be important for detecting the most relevant or salient stimuli in the environment and interpret those in relation to your internal states to guide behavior. So if you think about it, the default mode network, actually we know that it's not completely established until a couple of years, at least after 2 years of life. This network here seems to be one of those that you would think ought to be there almost from the get-go, because it would seem important to know what to orient in your environment. And so babies obviously would need to know that. Okay, so what did we find? What we found here, again, doing resting state and choosing the insula, which happened to be the hub of that network, we found the non-risk carriers actually had much stronger activity with several other nodes of this salient network, including the lateral prefrontal cortex, and also not shown here, actually a little bit there, the amygdala, and so on, than the risk carriers. Interestingly, when we again extract the values to get a different representation and look at the different groups, you see here in red is the typically developing and in blue is the kids with autism, that the toll that having the risk allele takes, it's greater in the kids with autism than it is in the typically developing. So having just one doesn't seem to matter in this case, unlike what we have seen for MET. I mean, if you don't have the risk allele or if you have just one, there are no significant differences between the two groups. But when you do have those both the risk allele when you're homozygous, if you are autistic, you do take this network is more severely impacted. So one of the interesting questions that we have been wondering about in this days is like, well, okay, we're getting impatient, so like, what else can account for this pattern of findings? And one of the things that Liana decided to do, since there are actually a number of different polymorphisms on this oxytocin receptor gene that have been implicated in autism, and we have data on all of them, is to actually look at whether the number of risk alleles that a child has on the same genes is related to social impairment. So what I'm showing you here is just a very nice, significant correlation between the ADOS communicative and social subscales indexing autism symptom severity and the number of oxytocin risk alleles that a child had. And as you can tell, okay, it may not be very steep, but we had a large N, and so this is a very nice, strong relationship. So I think this is like an indicator that we kind of need to look at on a variety of contributing risk factors, and the concept is that you may really need to take multiple hits to develop symptoms that are strong enough to warrant diagnosis. Okay, in wrapping it up, I have here just like a silly graph plotting some of the prevailing hypotheses that have been put forward in autism, and above here you see two that have been proposed and discussed at the behavioral level, if you will. So this mind blindness, this inability to understand other people's intention and their mental state and so on, and this lack of a social motivation that I've alluded to before. And I hope that even if I went very fast and I didn't quite develop the points as thoroughly as I would have had more time, that you may have seen that some of the neural system that I've talked about today may do a good job at accounting for this behavioral manifestation of autism and provide a kind of a neural mechanism that can account the neural correspondence for those behavioral hypotheses. We've also seen, although I think you can relate to the notion that if you have trouble understanding others and figuring out what their facial expression means, etc., that you may not really be eager to be socially interacting with them, because it would make it really hard. And likewise, if you're not interacting with them, I mean, you may obviously not be picking up, or be good at picking up those cues that would allow you to make inferences about what they do. So everything is quite interacting. The direction of these areas can rapidly change. We actually have some data that shows that these two systems are also, the functioning of these two systems is related. I've shown you, I think, some evidence, and some I did not have a chance, that shows that abnormal connectivity does relate to the functioning of this system. So one could just pass the ball and say, as I said earlier, well, this system are not functioning because those regions of the brain are not connected properly or the same way. And as much as there is no room, there wasn't room there for me to add, that I should also have another thing that implicates, with a bunch of autism risk gene. But the point is that, as I mentioned earlier, behavior impacts brain activity and as well as connectivity. And so we are left wondering to what extent all of this altered pattern of activity or connectivity are either causal or more the byproducts of having the disorders. And I don't want you to be leaving here today thinking, oh my God, this is all for nothing, and maybe we should just go home and give up. Because there actually are a number of things that we can do to tackle this issue about causality. Neuroimaging and genetics are ultimately correlational in nature. I mean, with genetics, you can go to the mice and you can actually do some things that you cannot do with imaging in people. But I want to leave you with the notion that all of this research in the past 15 years have actually been important for informing, in part, the development of treatment and interventions. And the most successful, dare I say so, and maybe Connie will take issues, but actually I hope not, are in a way have been fostering, or they work in ways that may boost both of this mirroring mechanism as well, make social interactions more enjoyable. So regardless of why you see some dysfunction in those systems, the notion is that you can still intervene and make them function, make those systems function more properly. But back to the causal and effect relationship, one of the things that we can do to address that is to really start doing longitudinal studies. There are I don't think that there is one single longitudinal study in autism. We have not followed these children, and at UCLA, that's what we are planning on doing, because we had the luxury of being funded by NIH for 10 years or more now. At least we've been scanning children for that long. So you can see how following the children along may allow us to disentangle some of what may be to identify alteration that may reflect true delay versus a truly deviant pattern of activity or connectivity. It's important also, and this is it's something that has emerged in the past few years as being quite important, to look not just at children with autism versus typically developing counterparts, but also to look at first-degree relatives of children with autism, say either their siblings or their parents, because we can, again, that way, this is a way in which we can highlight what carries risk, what is associated actually with the symptomatology versus what may be reflective of compensatory mechanism. We also need to continue to look at multiple autism risk genes, and ultimately I'm going to end with what I say the latest challenge for the UCLA imaging group, which is to actually push down the age at which we study these children and brain development and connectivity, and by studying infants who are at very high risk for developing the conditions, in hope to detect a signature for the disorders way before it could be diagnosed based on behavioral manifestations, and hence to start very early treatment like the one that Connie Kaiser is now implementing in babies at a high risk starting at 12 months of age. And I just want to leave you with some pilot data that we have collected just a few months ago to show you that in an 8-week-old baby asleep, we can actually do a very good job at identifying many of those same networks that I've shown you before being identified in adults. Not just the basic auditory or visual system or sensory motor, but also that salience network that I told you, it's my little personal pet, is the one that I would be willing to bet some money it's going to tell us something interesting. And last but not least, just to give you a sense that while there cannot be awake and being asked to be performing a task, we can nevertheless do also of the more traditional or conventional fMRI, but presenting the asleep babies with, say, speech that it's either in their native language, English, the language they heard in the first 6 weeks of life and also in uterus, versus, say, Japanese, for this baby, a complete foreign language. And lo and behold, this is again data on one single baby. You see much stronger activity or responses to the language that they have known and heard versus one that was totally novel to them, and this is the significant difference. So in a way, we can actually look at the amount of learning that has happened, and there are now studies that have been done in newborns 2 days old showing already some of these effects. So with that, I will thank you for your attention, and I'll remind you that if you have a child with autism or you know anybody, we want you, and thank you again.